So far, we have looked at how we can predict properties of proteins and of small molecules. However, we didn't consider yet how we can make predictions for properties that involve both a protein and a small molecule. In this video, we are looking at how we can predict different types of interactions between proteins and small molecules. As I said, we are now interested in predicting different kind of interactions between proteins and small molecules. This means we want to use information about a protein and a small molecule, and we want to be able to predict in which way those two molecules interact. But why is it even relevant that we can predict such interactions for enzymes? On the one hand, proteins, and in particular enzymes, play a crucial role in cellular metabolism, and hence knowing the function of enzymes is important in medical and pharmaceutical research for the study of metabolic diseases and drug development, and this requires knowledge about the interactions of proteins and small molecules. On the other hand, enzymes also play an important role in biotechnology and bioengineering, for example, for the synthesis of drugs, chemicals, and biofuels, in food processing, or in the degradation of waste materials. For these tasks, it is important to find proteins with specific properties that are suitable for processing or transporting specific substrates. However, for the vast majority of proteins, their function as well as their kinetic parameters is unknown. To overcome those limitations, it is desirable to develop prediction models for predicting protein-small molecule interactions. For example, predicting potential substrates of enzymes or transport proteins, predicting the Michaelis constants Km of enzyme substrate pairs, which describes the affinity or the binding affinity between an enzyme and a substrate, the turnover number Kcat of an enzyme catalyzed reaction, describing the maximum catalytic speed of an enzyme, or the inhibition of a small molecule drug for a target protein. The reason why this is interesting is that when developing drugs, a common scenario is searching for a small molecule drug that inhibits the function of a target protein. On this slide, I'm showing you a typical prediction pipeline for those prediction tasks that involve protein-small molecule interactions. If we want to predict an interaction between a protein and a small molecule, we need a numerical representation of the protein and a numerical representation of the small molecule. We've seen previously how we can achieve this. For representing the protein, we can use pre-trained transformer networks with the protein amino acid sequence as the model input, and to generate a representation of a small molecule, we have seen that we can use the SMILE string to calculate numerical representations using either traditional fingerprints or graph neural networks or transformer networks. Here I listed graph neural networks as an example. Since we want to have a single numerical vector containing both protein and small molecule information that we can use as the input of a machine learning model, one way of achieving this is by simply concatenating both vectors that were generated using two separate deep learning models. This vector can then be used as the input for a machine learning model that is trained to predict the protein-small molecule interaction of interest. We have already discussed that gradient boosting decision tree models are typically well suited for such prediction tasks. This protein-small molecule interaction task can either be a regression task, such as predicting how much a small molecule drug inhibits the function of a protein, or it could be a classification task, such as predicting whether or not a small molecule is a substrate for an enzyme or a transport protein. If we look at this process again, we see that we first separately generate numerical representations for the protein and the small molecule, and then combine both information in a concatenated vector. We see that this means that we have no information exchange between the protein and the small molecule until after the numerical representations are generated. However, this might be a limitation, and we might be able to generate more meaningful representations if we allow the model to look at the other molecule for which we want to make a prediction during the whole process of generating a numerical representation. The reason why this is difficult, or at least not obvious to achieve, is that the protein and the small molecule are typically represented on different scales. We have seen that proteins are typically represented through their amino acid sequences when generating protein representations, and small molecules are represented on a much more detailed level by representing each atom and bond in the molecule. To achieve a model that can represent both modalities, the protein amino acid sequence and the small molecule SMILE string in the same input sequence, it would be desirable to use a transformer network that can process the amino acid sequence of the protein and the SMILE string of the small molecule in the same input sequence. To achieve this, we can look at transformer model architectures that have been developed in the last couple of years for processing different types of model input, such as models being capable of processing both images and text in the same input. For example, we could have a transformer network that can take both an image and a text as the input, and we could predict whether those two input modalities fit together. This could be, for example, interesting when we want to search an image database for an image that fits best for specific image caption. There exist many different options to achieve such a model, and we're briefly going to look at the broad ideas for 3 of these here. In the following figures, the tokens of the 2 different modalities are represented by purple and green. As we have seen on the previous slide, those 2 modalities or input types could be, for example, image and text or protein and small molecule. One option of achieving such a model is simply concatenating both input sequences and using the resulting sequence as the input for a single transformer network. This requires that we tokenize both input types using the same vocabulary. This means that we have separate tokens for each input type, but they are stored in the same vocabulary and used as the input for the same embedding functions. This allows the transformer network to look at both input modalities simultaneously. A related option is to first pass each input type through a separate set of transformer network layers before using the embeddings of both sequences as the input of shared transformer network encoder layers. The advantage here is that we allow the model first to generate good representations of the tokens of both sequences before we allow the model to look at the other input type. It might be easier to extract useful information from the other input type once we already have meaningful representations. And the last option we're shortly discussing is using cross-attention between both input sequences. This means we have 2 separate sets of transformer layers, but for calculating the attention scores, we're using the embeddings from the other modality. We know that we have 3 types of inputs to our attention function: queries, keys, and values. In the image represented by Q, K, and V. And typically in our encoder, these are the same and are equal to the embeddings of the input sequence. But in cross-attention, we are also using the embeddings from the other input sequence to calculate the attention scores. This, however, has the disadvantage that we are not using any self-attention. And we can have, of course, also have completely shared layers on top of those cross-attention layers. We are now looking at the ProSmith transformer network encoder that allows to process both a protein amino acid sequence and a small molecule SMILE string. And the architecture of this model resembles most closely the second architecture type that we have seen on the previous slide. We first tokenize both sequences separately and pass them through the ESMB protein transformer network and the Chamberter 2 small molecule transformer network respectively. This gives us meaningful embeddings for all tokens from both input types. These tokens can be mapped to the same embedding space with the same dimension using linear layers. Since we have then embeddings of the same dimension, we can use the amino acid tokens and the small molecule tokens as the input of the same transformer network encoder. The attention mechanism of this transformer network now allows us to look at the small molecule while extracting protein information, and vice versa to look at the protein while extracting small molecule information. This transformer network can be trained for protein small molecule interaction tasks of interest, for example, for predicting whether a small molecule is a substrate of an enzyme, or for predicting how much a small molecule drug inhibits the function of a protein. For these prediction tasks, we have a classification token that you can see on the very left that we can use as the input for a fully connected neural network in the same way as we have seen for how we fine-tune pre-trained transformers. After we trained this model for predicting a protein small molecule interaction, we can use this as our final prediction model since we trained it already for the task of interest. Alternatively, we can also extract after training for each molecule or for each protein small molecule pair the updated classification token embeddings. This vector is now a task-specific representation of the protein small molecule pair because we trained the network to store all information that is relevant for the prediction task in this vector. We can then use this vector as the input for an alternative prediction model, such as a gradient boosting model, which we train to predict for the prediction task of interest. This typically leads to slightly improved model performance.